Там включим, здесь выключим.
Okay. Большое спасибо. Thank you very much for waiting. Thank you very, very much for this nice introduction. I'm very, very happy to be here as, scientist or professor of bioinformatics from Halle University of Germany, but also as member of this mirror lab here at Novosibirsk State University and at your Institute of Cytology and Genetics. So thank you very much for this wonderful time I have here. Thank you very much for this absolutely fantastic collaboration. And we just started, as Victoria said, we just started half a year ago. So what I'm telling you today is not the work that we did in the lab just in the half a year, but that is in fact work we did in the past. It's work on the phylotranscriptomic hourglass of plant and animal embryogenesis. But some of you may have heard a similar talk I gave here 3 months ago. And for those of you, I can say that the beginning will be similar, but the end will be new. Why is it new? Because we continued to do some work, and we found something quite interesting that I'm not going to reveal right now, but I can only tell you that we found something that even goes beyond embryogenesis. But let's start with this one. I would like to thank my dear collaborators. This is most importantly Marcel Quint. He's I would say, in the meantime, very well-known biologist, a biologist from Halle, one of the experts of auxin regulation, but he also got interested in evolutionary developmental biology. Then it is 2 of my PhD students, actually it's 3 of them, Georg Drost and Alexander Gabel, but also Markus Bönn. Carsten Ulrich is a postdoc in the lab of Marcel Quint. So this work was conducted at multiple institutions. On the one hand, at the Leibniz Institute of Plant Biochemistry at Halle, and then at our university. At the same time, we are also a member of the German Center on Integrative Biodiversity Research, and I'm quite happy to say that part of the research we do here in our mirror lab has to do with biodiversity research too. All right, so one fundamental question that some people on our planet ask, and I was only one of them, is, well, how does complex life emerge? And we are very interested in that question because of 2 reasons. Number one is, well, how did it emerge on the evolutionary level? But the other is, it emerges over and over again. Every time new life is born, biodiversity emerges. It diverges on the developmental level. And that is something we would like to understand. Our planet is full of complex life, and we would like to understand how could it happen? How could it evolve? And how does it, let's say, develop again and again each time from a fertilized zygote? Well, there's another question, and this has to do with biodiversity that we see on our planet. And what I mean by this is that the first question would be quite interesting, even if there was only one species, even if there was only one type of life, one species on the whole planet, even then it would be interesting to understand how could it have become complex by evolution, and how does it become complex over and over again in development after fertilization. But the interesting point is that on our planet we do not only have one species, we have more. We actually have more than 2, we have more than 10, we have actually many. And the question is, how does this biodiversity emerge? And again, we are interested in understanding that on the evolutionary level on the one hand, and on the developmental level on the other. Well, the interesting thing that you can see is that embryogenesis has emerged 2 times in evolution on our planet. Bacteria are not capable of, well, complex life form that we call embryogenesis, but plants and animals undergo embryogenesis in their development. And this is exactly the reason why we are interested in studying these things. Comparative embryology is one term that was used 100 years ago. Later, we call it evolutionary embryology, or today we just call it Ivo. So I would like to tell you a story that is really interesting. So there's this historic figure, this pioneer of science. I mean, he was a universal scientist, he was a medical doctor, at the same time he was interested in well, biology as we would call it today. Of course, at those times, he was also a great artist, wrote poems and so on and so forth. But this person, even though he worked in Germany, and you believe Germans are always very precise, they document everything they do, he made a crucial mistake. And it's one of the mistakes that we try to teach to our students not to do. And this is, if you do experiments, then write down very precisely what you did, label your data accordingly, and so on and so forth. And despite we teach this, it just happens that samples get mixed in the refrigerator, or we send something to a sequencing center, we get the samples back, and then we find, oh, something was confused in the sequencing center. What they call control is actually treatment, and what they call treatment is control. This still happens, and interestingly, it happened to this famous scientist. So at some point, what he did is he was interested in embryology. So he took small embryos of different developmental stages, of different species, and he put them in alcohol to preserve them. And at some point, he wanted to look at some of the species, and he looked at these two glasses, and he found out, I cannot distinguish them anymore. I don't know what they are. And this is when he made a famous discovery. And I try to read this to you. This is old-fashioned German, so some of you guys who know German should not be afraid right now that your German is bad. Even Germans can almost not read this because this is German language from 200 years ago, which today is hard to comprehend. But I like to read this to you in English language because it's a quite interesting story. So he says, I have two small embryos that are preserved in alcohol, for which unfortunately I forgot to write down the names. Well, and now I'm not capable of saying what the class of these two species is. It could be lizards, it could be small birds, or it could be young mammals. This is how similar their anatomy is. So the point that he discovered by miracle, by making a mistake, he made a great discovery. And this is that animals or embryos of different species, they can look very similar at early stages of development. So originally it was stated a little bit differently. It was stated in this original work, I think of 1828. It was stated that embryos of different species progressively diverge from another during ontogeny. So today we can say that the definite finding is that he discovered there is a stage in which embryos of different vertebrate species could not be distinguished. Well, this today we know is one of the milestone discoveries of developmental biology. All right, the interesting thing here is that this was before Darwin. Nobody knew about evolution. And let's see what happened then in the history of biology. Well, the origin of species were published, so Darwin was the person who delivered the connection of the theory of evolution on the one hand and the common origin of species on the other. By which mechanisms? Of course, we understand it today, by natural selection. But the question was open at Darwin's times. If there was any connection between the origin or descent of the organisms on the one hand and systematics or ontogeny on the other. And there was another German scientist that entered the stage. His name is or was Ernst Haeckel, and he formulated this famous biogenetic law. He says the ontogeny is the short and fast recapitulation of phylogeny. Now what is important about this discovery is that today it's considered mostly wrong, and unfortunately it's true that it was based on purposefully falsified data. Moreover, later, Nazi Germany abused these findings for their terrible racistic ideology, but that, of course, is not the fault of Haeckel. The good point, however, is that Haeckel was the person who, for the first time, discovered that you can approach developmental biology from an evolutionary perspective. All right, so what do we have? What do we have in the 19th century? Well, we have, on the one hand, the contribution by von Baer, who said that there is a stage of maximum morphological conservation during embryogenesis of different vertebrate species. And then Haeckel was the person who said, well, you can integrate these different disciplines, such as taxonomy and embryology, into the new Darwinian framework. So, if we summarize what we learned up to now, if we summarize what mankind knew in the 19th century, then this can be formulated as the hourglass model. The term hourglass model was coined only later, but people understood at this time that in mid-embryogenesis, there's a stage, and they call it phylotypic stage. There's a stage when, you know, the morphology of different species is quite conserved. But at the beginning of embryogenesis and at the end of embryogenesis, you see diversity. So in terms of time progression, zygotes of different species start to be quite different, and then they develop. And as they develop, they become more and more similar to each other until they hit the stage where they are almost indistinguishable from each other. And only afterwards, diversity or biodiversity increases again and literally explodes, so that at the end of embryogenesis, we can clearly distinguish an elephant from a giraffe, from human, from monkey, from cow, from dog, and so on and so forth. All right, so that was the stage in the 19th century. And of course, a lot of progress has been made, or had been made since then, but a pioneering, well, let's say quantum leap happened in 2010. There were 2 papers in Nature published back to back where people found a molecular explanation for this strange hourglass phenomenon. And the molecular explanation is the following. It was by this group found in zebrafish, by the other group found in Drosophila, and the evidence is the following. If you look again at temporal development and pay attention, now time goes down, was not my choice. This is what the authors chose. At time, the zygote is here, and development goes down. So the hourglass, you know, is just on the top. What they noticed, and let's just look at the right part of this figure, is that at the beginning of embryogenesis, the genes that are expressed are relatively young, in terms of evolutionary time. In mid-embryogenesis, however, it's mostly ancient genes that are expressed. And at late embryogenesis, again, it's young genes. So that could be one possible explanation on the molecular level for this morphologic hourglass. Because if at the beginning of embryogenesis you express young genes, well, young genes by definition are different from species to species, well, then perhaps the phenotype should look different. If in mid-embryogenesis you express old genes by definition are those that are similar between different species. Well, if you express these old genes that are similar from species to species, well, then maybe the phenotypes should look similar. And then at the end of embryogenesis, if again you express evolutionarily young genes, young here means they are different from species to species, then it's not surprising that the phenotypes at the end of embryogenesis look again different from each other. So, the interesting discovery was that this stage, this developmental stage, where the oldest genes are expressed, coincides perfectly with the phylotypic stage that people knew from morphologic studies. All right, embryogenesis. And this is that at the beginning, oh sorry, here's the beginning of embryogenesis now. Young genes are expressed. In mid-embryogenesis, it's old genes. And then at the end, it's again young genes. That was the state of 2010, or let's say 2011, because the papers were just published in, I think, the last week of December, year 2010. And our question at that time was, okay, people have discovered this for an insect, an invertebrate, and they have discovered it for a vertebrate, for zebrafish. What could be the situation in other species? And we were not only interested in finding out, well, what could be the situation in other animal species, but we thought, what about plants? And the reason why we were particularly interested in plants is the following. Plants and animals, of course, have a common ancestor. This approximately is the common ancestor. It lived about 1.6 billion years ago. And one thing is clear. This common ancestor was unicellular. So it's clear that in this unicellular ancestor, you cannot have any genes that have some purpose or some function for embryogenesis. In fact, look at this plot. It's actually taken from one of the classical papers of Elliot Meyerowitz. And what you see here is that approximately 1 billion years passed, 1 billion years of evolution passed in both the animal lineage and the plant lineage before in both lineages multicellularity evolved. So it took about 1 billion years in both of these lineages independently of each other to evolve multicellularity. And then it took another time, which I cannot fully specify, but at least another few hundred million years until, again, independently in animals and in plants, embryogenesis evolved. So at that point, it was really interesting for us. If there is this morphologic hourglass in animals, which must have evolved somewhere here. Somehow at this point, this hourglass must have emerged. Then the question is, what's going on in plants? Because it's an independent process that we could study here. So the question is, well, would we perhaps see a similar pattern in plants, or would we see a totally different pattern? Well, let's look at what we need to do this study. So in order to perform phylotranscriptomics, you actually need only two pieces of information. The one thing is you need to have the evolutionary age for each gene, and the other is you need to have transcriptome data. Transcriptome data of different embryo stages. Okay, well, how can you get the evolutionary age of a gene? I mean, genes don't have a passport, right? You cannot just open the passport and see, oh yeah, that gene originated 560 million years ago and the other one is younger. Impossible. So this is something where you need to perform some work, where you need to do some bioinformatics. But one approach that actually works, at least in principle, is called phylostratigraphy. And this is that you look at a phylogenetic tree of interest. So usually it's the tree, which is called a ladder tree, where you say, okay, you have your species of interest, in our case, Arabidopsis thaliana, one of the nodes, and then you go through these intermediate nodes that correspond to larger and larger phylogenetic sets. Until in the end, you come here to to the root node, which is just all life. And the question is now, for each gene, for each of the 30,000 genes of the Arabidopsis thaliana genome, we would like to know how old these genes are. And how do we define age, or how is age usually defined in this area? Well, very easily. If you have a gene that has no homolog, if you have a gene of Arabidopsis thaliana that has no homolog anywhere else, well, then you say this is an Arabidopsis thaliana specific gene. So perhaps it emerged only in the last, let's say, 5 or 10 million years since the last common ancestor with this sister plant Arabidopsis lyrata. Yeah, if you don't find this gene of interest even in Arabidopsis lyrata and you don't find it anywhere else, then it's a very young gene that's thaliana specific. Now in contrast, if you have another gene and you find homologs or or I should say orthologs, everywhere. You find it in different plants, you even find it in animals, you even find it in prokaryotes. Well, if it's such a gene, then you say that's a very old one. That actually will be grouped in this category. That can be found in prokaryotes and eukaryotes. And, you know, if there's an intermediate gene that you find, let's say, in the group of all rosids, yeah, you find it in the group of all rosids, but you don't find it anywhere else, well, then you say this is a like half-old gene, and that is then grouped in this age category corresponding to rosids. Well, people do it in a simplified manner. We just call these different groups phylostrata, and this is an idea that's taken from geology. In geology, people drill in the earth, they have different strata, and they know the deeper you drill, the older your layer or your stratum is. And the same idea was used here, pioneered by Domazet-Lozo and colleagues in 2007. So just for every gene, you do a blast comparison to all other genomes that are available. You look, where do you find homologs, and according to the set of homologs you find, or the, let's say, the phylogeny of the species in which you find homologs, you group your gene into this one of these particular phylostrata. So in our case, phylostratum 1 is the oldest one. It corresponds to all life. And phylostratum 13 is the youngest one. It corresponds to Arabidopsis thaliana specifically. So basically, as I said, you just perform a blast search of your one gene against all sequenced genomes. You get this age category of your gene, and you repeat this step 30,000 times for all 30,000 Arabidopsis thaliana genes. That's the idea. So I just summarized it here. So of course, on the bioinformatics level, you construct your tree, you define your phylostrata, you construct a database with all sequenced genomes, you blast each gene or each protein of the species of interest to this database, and then you sort the genes into these phylostrata. Okay, the result is something that is called phylostratigraphic map. Basically, it means that for each gene in the Arabidopsis thaliana genome, you know the age. Okay, first problem solved. Yes, I know it takes some time. I think it takes more than a CPU year of compute time. But on the other hand, all of us have powerful computers nowadays, and at all institutions you have compute clusters. And these compute clusters are quite happy that they can compute meaningful things and not just burn energy. So on a cluster, that's maybe a job that runs for a couple of weeks, and that will be it. Okay, so we got this phylostratigraphic map, and then comes the next step. The next step is, well, how can we, use this information and combine it with transcriptional data? The answer is relatively straightforward. So you just compute something that's called the transcriptome age index, which is just the mean evolutionary age of a transcriptome. Now, what does it mean, mean age? Let's assume expression data were binary. Binary means a gene can either be on or it's off. This is a simplified assumption. We will relax that assumption in a second. But let's assume someone told us, oh, genes can be either on or can be off. Well, then it's very easy. I take my transcriptome, I look at all the genes that are on, I ask each of the gene, how old are you? And I take the average age. That's it. It's exactly how we compute averages everywhere in life. The only difference here is that we say, let's take the weighted average. So if a gene, for example, is, let's make it simpler for you, if it's on, you count the weight as 1. If it's off, the weight is 0. But there may be a gene that is half on and half off. Well, in that case, you would just give this gene a weight of 1/2. And now in reality, of course, we have numeric values for expression data. Largely expressed genes get a higher weight than lowly expressed genes. But that's just called a weighted average, and I think it's also sometimes, or it's something that we often do. So anyway, this transcriptome age index is just defined as the weighted average of the ages, where the weights are just the expression levels. All right, so we get this transcriptome age index, every transcriptome for which we have expression data. All right, so we want to now compute this transcriptome age index for different transcriptomes of different developmental stages in embryogenesis. That's our goal. That's exactly the goal we have here. And then we want to see what this profile looks like. Okay, that's the expression data that was fortunately taken right at the time when we needed it. You understand that measuring expression data from these tiny embryos is technically extremely challenging. That's exactly the reason why there's so much data out there on zebrafish embryogenesis, on even Drosophila embryogenesis, and so on. But in plants, it's much more difficult. So at that time, it was in year 2012, there was only one single such data set that existed in the world. And fortunately, we live in a time where every one of us is happy to share data, to share results, to share insight. So we were quite happy that this group from Canada had taken and measured these data. They used the data for their purpose. They were very happy to share it with us. And so we could use this data set to look now at the transcriptome age index at these 7 different developmental stages. That's what comes out. And again, in this case, the hourglass or the time goes from bottom to top. And high values of this transcriptome age index, remember, high values of phylostrata means young groups. Small values of these phylostrata mean old groups. PS1 is the oldest, PS13 is the youngest. So what we see here is that at the beginning, we see in plants, we're not in animals anymore. And there shouldn't be any reason why there's an hourglass pattern in plant embryogenesis. But that's something we see here, even if we don't like it. But nature shows us that there's one. At the beginning of embryogenesis, it's relatively young genes that are expressed. They get progressively older until they reach in the torpedo stage, a stage with the oldest transcriptomes, and then afterwards you can see they become younger very rapidly, so the youngest transcriptome is in fact that of the mature stage. All right, let's summarize up to now. The intermediate conclusions are that embryogenesis evolved twice and independently in animals and in plants. Embryogenesis is morphologically and genetically very different in animals and plants. I didn't talk about it in detail because I assume you know about this much more than I do. But the interesting thing is that transcriptomes of both animals and plants show the same hourglass pattern. So at this time, it's not really obvious of what the reason could be. Why should there be this universal hourglass? That is a question we like to address. But before doing it, we like to address the question, is this hourglass that we observe an evolutionary relic? Maybe it was functional and important 300 million years ago, but it's not important anymore. We just see a relic. We just see a shadow of some pattern that was maybe much stronger 300 million years ago. And it's fading away, but it's fading away slowly, so we still see the remnants, the shadow of that pattern that was functional a long time ago. Or is it actively maintained? Is this pattern perhaps still of functional importance today in plants that exist today? So that's what we would like to study. Of course, you understand if you address these evolutionary questions, very often it's not possible to do a direct experiment. Often you have to combine different independent pieces of evidence, and that's what we are going to do. So I'm not saying that this will be now an experiment that is conclusive, but I'm only saying that we try to do a set of, let's say, experiments or studies that will provide indirect evidence. And what exactly is it that we want to do? Okay, we would like to look at another index that you can compute from transcriptomes. We call it transcriptome divergence index. So that's based on the sequence comparison of orthologous genes in related species. So the sequence divergence, if you remember, is the ratio of these Ka and Ks values, the non-synonymous and the synonymous substitution rates. And well, then basically what we do is we use this KAKS value instead of the PS value, instead of this phylostratum. So remember, these phylostrata, they have to do with long evolution. They have to do with long evolution going back 2 or 3 billion years, to 2 or 3 billion years ago. KAKS, of course, only focuses on very recent selection, focuses on whether there's positive or negative selection, let's say in the last 5 or 10 million years. So it's interesting. It's like a microscope. You have different layers of resolution. With the classical tie analysis, you go back in evolution very long. With this analysis, you stay very short as you can stay with, some related species that you still need. So in this particular case, we chose these two species, Arabidopsis thaliana and Arabidopsis lyrata. These are the species that we take, we align them, we determine the Ka-Ks ratios from them, and given that the last common ancestor of Arabidopsis thaliana and Arabidopsis lyrata lived about 5 to 10 million years ago, it's clear that this measure, whatever we measure here, has only to do with very, very recent evolution ranging back at most 5 or 10 million years ago. All right, so that is the patterns we get. And again, zygote is at the bottom, mature embryos at the top, so time goes from bottom to top. This is the pattern we see when we do this comparison between Thaliana and Lyrata. So it starts at, divergent, let's say in early embryogenesis, we have a quite divergent transcriptome. It becomes more and more conserved until it reaches the torpedo stage. And then after the torpedo stage, it becomes again divergent. And if you think that this could be an artifact just of comparing Arabidopsis thaliana and Arabidopsis lyrata, you are in our team, because that's what we thought. We thought maybe this is only an artifact. So we also did several other comparisons with Capsella rubella, T. halophylla, Brassicarapa, and so on and so forth. And what you can see is that the specific shape of the hourglasses changes. But in all cases, it remains an hourglass, and in all cases, the stage that has the highest conservation is the torpedo stage. And remember, the torpedo stage was the phylotypic stage of plant embryogenesis, where we also had the highest, where the transcriptome that is expressed is the oldest. Putting both things side by side, you see this is the hourglass that we see when we look back in evolution long time, until 3.8 billion years. This is the TDI, the transcriptome divergence index, that can only look at very recent evolution and only looks at positive versus negative selection. And these two patterns are almost mirror images of each other, suggesting that this transcriptomic hourglass is in fact under active maintenance. It's probably still functional today. It's probably not only a shadow or a relic of an hourglass that was much stronger and functional only 300 million years ago. Okay, so that suggests active maintenance and thereby possibly functionality in Arabidopsis thaliana. Now that is an important conclusion, because if that was true, then we have a chance to make functional assays or functional studies in species that exist today. If it had been only a relic and is not functional anymore, then you could do no experiment in plants or species that exist today and learn anything about the function of the hourglass, if it had been only a shadow of evolution. All right, the next question, of course, is, nice, that's in plants, but what about animals? Well, this is something we looked at. These are now the other two species that I mentioned, zebrafish and Drosophila. These, by the way, are the hourglass patterns. So if some of you think, I'm not sure if this is an hourglass or not. Again, we agree. But at least these patterns were published by the other groups as hourglass patterns. So we should not be too critical here. And if we look now at the TDI, then we see again something that, well, is not maybe a good hourglass, but is, well, consistent with an hourglass pattern. The p-values are provided here, so they tell you to which degree these results are significant. So in conclusion, yeah, there are also significant hourglass patterns. In this case, maybe we could say it's only half an hourglass. And I can tell you, we looked, there are transcriptome data available for this part. This is post-embryogenesis. And it then goes up very highly. At least I think we can conclude this phenomenon that the hourglass pattern seems to be actively maintained is a phenomenon that seems to be not restricted to plants, but it seems to exist also in animals. Okay, so again, we are ripe for intermediate conclusions. Nature apparently invented embryogenesis twice and independently. As I mentioned, embryogenesis is very different in plants and animals, and the developmental hourglass seems to be essential. Why? Well, because it was invented 2 times independently, and if it's not essential, you could still speculate, well, this is by chance. But in an evolutionary context, if you find a pattern, it's very rarely by chance. It usually has a reason why it is there. So this is why we believe, well, perhaps it encodes some essential function, which we don't yet understand. And it seems to be actively maintained. And what that means is, it seems to be functional, and it seems to give us a chance to do experiments to find out about its function. All right, this is just a pattern that reflects it. So recall, there was the single-celled common ancestor. It took a while until multicellularity emerged in both lineages independently of each other. It took another while until embryogenesis emerged, again, independently of each other. But in both cases, we see this hourglass, where we can now summarize, is not only the old genes, the young genes that are suppressed in the middle, but it's also the divergent genes that are suppressed in the middle. All right, that is our second, let's say, conclusion. And now we were interested in learning something about other developmental processes. And the interesting thing is, if you compare organ development, or I should say, in general, development of animals on the one hand, this is a particular case of a mammal, maybe some of you recognize what mammal it is, and you compare to plants, then you see a very interesting difference. And this is number one, in embryogenesis, in animal embryogenesis. Organogenesis happens simultaneously. You have a simultaneous development of different organs, and it happens within embryogenesis. So some of my friends from animal embryology actually say, well, in our field, embryology and developmental biology is almost the same. Because at the end of embryology, we are ready. Nothing more happens in animals. We just grow. Yeah, we may become smarter. Some of us may not become smarter. Okay. But in terms of organ development, not much happens. I think it's only one organ, it's our, what is it called, sex organs that develop in a post-embryonic manner. Everything else is already in place. Now in plants it is totally different. If you would draw, on a morphologic scale, if you would draw embryogenesis on a same fictive scale, like from 0 to 1, then we would say in plants, embryogenesis is only the very beginning of development. In plants, you have a lot of post-embryonic development. Most of organ development in plants happens after embryogenesis, not within embryogenesis. And moreover, it doesn't happen at the same time. It happens step after step. And this, of course, makes plants a wonderful object for study. In many cases, when we in plant biology have a great idea or make a great discovery, we will see, in the animal field, it was found already a few years earlier. So there are many cases in which, non-plant biologists, like we call them animal biologists, are ahead of plant biologists. This is now a unique chance where we in plant biology can do a study that people in animals cannot do easily. And this is because in plant development, there are many developmental processes that come after embryogenesis, and organ development goes in sequential steps so that we can study different developmental transitions independently of each other, which would not be possible in animals because here everything happens at the same time. All right, so the question we are addressing last is, are transcriptomic hourglasses unique to embryogenesis? I mean, it was a major surprise to see that they're not restricted to animal embryogenesis. No, they also exist in plants. But now we like to ask the second question, the next question, and this is, maybe this hourglass pattern is not restricted to embryogenesis. Maybe it's even a more general phenomenon that we may possibly find in some of the other developmental processes of post-embryonic plant development. That's exactly what we would like to study. So let's look at the typical, life cycle of a seed plant. So on the one hand, you have embryogenesis. Well, I put a check mark here because it means this is what has been studied already. I just showed you the results. This is where we see an hourglass. So the next question is, germination is the next step that comes after embryogenesis. Let's see if perhaps there is or there's no hourglass in germination. Let's do this. This is a data set we took. This is hours after sowing. Here you see the transcriptome H index, and you see very nicely an hourglass pattern in germination. So the conclusion at this point is that not only embryogenesis has an hourglass pattern, also germination. So 5 years ago, people thought the hourglass pattern is something specific to animals. We could find no, it also exists in plants. Now it goes beyond. People up to now thought, including ourselves, well, this hourglass pattern is called embryonic hourglass pattern, so it's something characteristic for embryogenesis. Now we find no, it doesn't. It even goes beyond. It also exists in germination. Well, the next question was, what about floral transition? And this is something that I can only answer with words right now, because these data are very fresh. So floral transition is this stage where the plant switches from vegetative growth to generative, reproductive growth. And in this transition, again, we took data, we measured transcriptome data, we looked at the transcriptome H index, and the data I just got this week from my dear friend and colleague Marcel Quitt and the lab that we are running in Germany, is that also in floral transition there's an hourglass pattern. So, yes, it seems there are many hourglasses, and the hypothesis that we now start to develop is maybe it's a much more general phenomenon. Maybe it's not, we see it's not only related to animals, it exists also in plants, but it is also something that is not only related to embryogenesis, it's perhaps related to many different developmental processes. So von Baer and Haeckel were right. They talked about the connection of embryogenesis and evolution. But it is possible that this pattern that they found is actually only the surface of a much deeper pattern, of a much broader pattern that is not restricted to animals, is not restricted to embryogenesis, but it may be present, it may exist in a much wider area. So the checkpoint hypothesis could be that whenever you have crucial developmental transitions, then perhaps the cells, the organisms, are required to shut down young genes. We don't understand why, but it's a hypothesis right now. Maybe in these critical stages, it's of evolutionary advantage, of selection advantage for the plants, at least for the plants, to shut down young genes and activate the old genes. And then it would mean that in the life cycle of a plant, you see these constellations. You have multiple hourglasses. So the conclusions, and these are the final conclusions now, are yes, these developmental hourglasses seem to be essential, they seem to be actively maintained, they might still be functional, and there are post-embryonic hourglasses in plant in plants, both in germination and in floral transition. And that, of course, could suggest some, existence of a universal logic that coordinates embryogenesis in both kingdoms and several developmental processes in plants. It could be. We don't know. We don't know what this general process or general phenomenon could be, but there seems to be some general universal force that forces plants in different developmental stages to deactivate young genes in critical stages of development. Now that, of course, could then be the pattern that governs the emergence of complex life and biodiversity on our planet. And with that, I'm at the end, and I would like to thank my very dear colleagues from this German Center of Integrative Biodiversity Research. It's a wonderful center. I mean, we got funding for 100 million euro to set it up. It consists of 8 different professorships at these 3 different, university cities of Germany, located in 3 different states of Germany. It's a really fantastic research environment, and of course, within this research environment, we like to continue this research. And I should say at this point that in the same way in which I'm extremely happy to be member of this German Center for Integrative Biodiversity Research, in which we get the possibility of carrying out our research there, I'm likewise happy, equally happy to be here with you, to be here at this Institute for Cytology and Genetics, as well as at Novosibirsk State University. And for that, I would like to thank the people here in your country, which make it possible that scientists can collaborate despite some political friction, Your country and our country, at least the governments, sometimes don't understand each other, sometimes disagree with each other. We may disagree with the German government. We may disagree with the Russian government. That doesn't matter. What matters in science is that we collaborate, that we are friends, that we work together and not against each other. And this is the mission we want to carry out through our Mirror Lab. And for this possibility, I would like to thank all of you, and I'd like to thank you also for your attention. Thank you.
Okay, questions.
Ivo, how do you think these new genes and conservative genes, if you see the difference in points of expression, and you know that there is a genetic background of difference between different species. Yes. And there may be some conservative core.
Yes.
Making the development processes stable. And some new genes impacting differences, shaping the form of organisms under development. How do you think? Is it so? Did you find some conservative core making the process of development stable, at some point stabilizing it, and other parts are just the parts contributing to differences, to special specificity? Or it's not?
It's an excellent question, and I should give you an honest answer. We haven't found it yet. But it doesn't mean that it doesn't exist. It only means that perhaps the analysis that we have done were not deep enough. And I should say that we tried to look at what you suggest, a little bit, but I should be honest and say we looked at this only a little bit. And I'm sure that we should look at this in more detail and deeper, and then perhaps we find exactly what you suggest. So the honest answer as of now is we haven't looked deeply enough, we didn't see any indication, but it doesn't mean anything. And I can tell you one thing that we found, and this is that if we go back, let's see, where do we have an hourglass? Well, there are many. For example, here. We asked the following question. The genes that are active here, these young genes, they're downregulated towards the phylotypic stage, and they're upregulated, and afterwards we see an upregulation of young genes. Well, What I didn't tell today in detail, because I wanted to stay within the 45 minutes, is that we did a detailed analysis. I think I showed it to those of you who could come to the previous talk. I showed it in summer, that we looked specifically at old genes and at young genes separately, because we wanted to find out if the origin of this hourglass is the, let's say, temporal change of expression of the young genes or the old genes. It could be both, right? It could be that young genes are active here, they are downregulated and they are upregulated again. But it could also be that young genes are expressed in a constant manner, and it's the old genes that are downregulated here, they're upregulated in the middle, and they're downregulated again. So your intuition was very good, or you attended the talk in summer, I don't know. But you guessed it correctly, that the old genes are expressed in a relatively constant manner, and it's the young genes that are responsible for this hourglass pattern. And now we ask the question, are the young genes that are active here the same as the young genes that are active there? And it's not the case. Some of them are, but the set of young genes expressed here and the set of young genes expressed here doesn't overlap strongly. So the more precise description of this hourglass pattern is, there are some evolutionary young genes that are apparently important at the beginning of embryogenesis. They are downregulated towards the middle. And then it's mostly other young genes that are then needed for late embryogenesis that are upregulated here. So this is just additional indication that the study you suggest could be extremely valuable. So thank you for this very good question.
Thank you very much for a great talk, as always. So your hypothesis is that you have controlled oscillations and controlled inhibition of the expression of the young genes. But might it be the case that you have uncontrolled oscillations which are going on all the time, and in fact it is a chaotic, so at the end it's the result of not chaotic but uncontrolled oscillations which are happening in the gene expression system which are responsible for this part?
It could be. Yeah, you are very right. I mean, you talk about oscillations. Maybe I use the word, but usually what we understand by oscillation is really some, harmonic, periodic thing. It's very difficult to describe it here because we have these different developmental transitions, and of course we can measure only a few time points before and after because experiments are still very expensive. So theoretically speaking, you are absolutely right. We don't know if it's a periodic or if it's quasi-periodic or chaotic. We don't know this, and it could be that it is chaotic. But I think it will be very hard to measure it, to get an answer to your question by experiments, because we can really measure only a few time points. And the only pattern, the, let's say, rough description of the pattern that we like to give is that we say, well, it goes down, it goes up again. If this is part of an oscillation, of a periodic oscillation, or if it's part of a quasi-periodic oscillation, or if it's even only part of a trajectory that is chaotic, we don't know, and we will try to not speculate about it. Yeah, I mean, it's always hard when you write a paper, you may talk about oscillation, you don't think as deep as you did about is it a real periodic oscillation or is it chaotic. My feeling is we will not be able to answer the question, and then the question is what is the most simplest assumption, and I wouldn't say fully periodic, but maybe the term oscillation is okay in the sense that it's high, low, high. Maybe on this simple level, maybe it's only this simple level at which we can describe the pattern. But great question. So perhaps we can, perhaps it is possible to study other things than transcriptional response, for which measuring, getting data is much, much cheaper. And maybe we can look at these dynamical processes. I know you are particularly interested in a few, and it is possible to look at these and then see if there's, constants or there's something like oscillation. And perhaps there it might be possible to disentangle or distinguish between regular oscillation or periodic oscillation and chaos.
Hello, Dima.
Okay.
Войди сюда.
I wonder how broad were the genes that have expressed much in the low evolution, high interested in research of seed development and development of the flower, because these two organs are quite new in plant. And did you observe the expression of very old genes that are introduced in plants before the seed or flower appears in plants?
Yeah.
And in this way, how they function may be related to development of such quite new organs.
Yeah.
These genes are really very old. So why should they express in this stage?
Okay, why we don't know, but I can answer your first question, and this is how old were the genes. So you're referring to these genes that are expressed, for example, in the torpedo stage. And unfortunately, I didn't bring detailed plots here, where we also looked at the expression of genes of these different phylostratigraphic groups. And I would like, from this picture, I would like to jump back now to this phylogenetic tree and show you what the old genes were and the young genes. So let's go back, to this one. And I can tell you that the old genes, well, the genes that we call old genes, are those that belong to the first three phylostrata. Only the first three. That was the old ones. So there's cellular organism, eukarya, eukaryota, and viriplati. And all other ones that come afterwards, we declare as young. And there's a reason for this grouping. So this.
I just want to understand. Is it? I mean, what you call the old one? Is
Yeah.
it not? It's not a housekeeping genes. Or
No.
it is also housekeeping.
Well-
or only just developmental genes. Because it's housekeeping. I mean,
No,
are they there or
no,
not?
it's housekeeping genes versus non-housekeeping genes.
Yeah.
And old genes versus young genes is a different thing.
Right.
Because housekeeping only has to do with expression profiles,
Yeah.
that it's relatively constant.
Ah.
And old versus young
So
only
they are
has
not
to do.
considered here.
Well, the housekeeping genes are part of it. Let me give the
Right.
full answer. I'm only saying these are two separate categories. Housekeeping versus non-housekeeping has to do with expression data.
Yeah.
Old versus young, as in this context, has to do with only with phylogeny and homology or orthology to other species.
But
I
most
can-
housekeeping are very old.
Exactly, that's what I wanted to point out,
Yeah.
that most housekeeping genes are old
Yeah.
and most old genes are housekeeping genes. Okay, yeah, that's what we found.
Yeah.
But not all, most, yeah?
Right.
And now come back, what is old? And in this particular case it's interesting because we found, so to speak, a striking difference of the dynamics of expression between genes that are in groups 1 to 3 on the one hand, and genes that are in groups 4 to 13 on the other. And the striking difference is that genes in groups 1 to 3 are expressed in a relatively constant manner, and genes expressed in all of the groups 4 through 13 are expressed in a way that it goes down to mid-embryogenesis, reaching the minimum in the torpedo stage, and then go up again. And now look at the evolution. Genes, gene groups or age groups 1 to 3 are those of, let's say, groups of species before embryogenesis, and the other groups 4 through 13 belong to species that all undergo embryogenesis. So that is the only insight we have, but we don't understand why, to be very honest. The why question would be, why is it these young genes that evolved after, evolved in species that undergo embryogenesis, why are they downregulated towards the middle and upregulated again? Or to come back to Alexei's question, why is it that, some young genes are up here, downregulated, and some other genes that are also young are then upregulated again? We don't know this yet, but I hope I could answer your question of how old are these old genes, these, predominantly housekeeping genes. How old are they? Well, they fall in these first categories, and we didn't put times here because time is debatable. But you go to timetree.org or anything that gives you consistent, estimates of the times, and then you will find out how many 100 million years ago they were.
Thank you.
Great question. Yes, thank you. More questions?
Hello, Roland.
Thank you for the talk. Could you please specify about house genes? Are they old or new?
I cannot. I because all of my talk had to do with Arabidopsis thaliana, and of course Hox genes are mainly studied in animals. So that is something we can discuss afterwards, because it may have to do with only the one slide on which we studied the active maintenance in Drosophila and in zebrafish. But here we looked at the Ka-Ks ratios, right? At the positive and negative selection, and not at the original, let's say, transcriptome age index that looks at evolutionary age. But perhaps you find the answer, or both of you and I afterwards, when we discuss, can look at the original Nature paper of Domazet-Lozo and colleagues. Maybe the answer is already given there.
It's supposed to be a lecture for young scientists. Let us also ask questions. So, if any common mechanisms for gene expression regulation for wet genes that are downregulated at the middle stage, so it may be related to chromatin organization or some common mechanisms, all of them gene targets or some transcription factors, so maybe some condensation of
You are absolutely right, it could be, and we tried extremely hard to do these analyses. And I should be honest again, we didn't find anything. So we looked extremely hard at different GO categories, at the genes that are, downregulated towards the middle, and at those genes that are upregulated from the middle. We did really extensive studies. I mean, not only a few days, but it was hundreds or even thousands of hours that we spent on looking at different functional categories, at different location on the chromosome, at all possible features we could look at, we tried to look at. And unfortunately, up to now, we didn't find any consistent pattern. So sometimes it is that you think, oh, there's a functional category overabundant, and some others may be underabundant. But if you do a rigorous, statistical test, or you vary your input data a little bit, you do bootstrapping analyses, then all these, let's say, suspicious patterns disappear. So honestly, I must say, Yuri, that up to now, we haven't found anything. We would love to. And it's clear that, we haven't done all analyses that could be done. And this is exactly why, we need your help too. We need the help of mankind, that this is probably only solvable by multiple groups, in the world, in this country here, but also in the rest of the world, that, have to work together on trying to understand why it is and what the regulatory process is. So we also looked at transcription factor binding sites and other things. Unfortunately, up to now, we didn't find anything that's significant.
Well, there were in fact 3 cases of multicellularity. The fungi.
Yes,
What about them?
Okay, excellent. You asked excellent questions today. I told you, I'm so happy to be here, and I'm also so happy to be here now in this talk and in this discussion with you. So, in terms of the first slide, multicellularity evolved really multiple times. Even some bacteria can be stated to have multicellularity.
And animals, I think it's not just once.
Yes, exactly. So multicellularity, I think, is only one thing. Embryogenesis was what we were interested in the first place, and here, of course, we have to state it's only 2 occurrences, once in animals and plants. But fungi are a very interesting subclade, and there has been a publication this year by a team of people who studied fungi development, and they also discovered an hourglass there. So that is really interesting. So you had a great intuition. So these people.
Well, when you know what to see, it's quite easy to see.
That's right.
It can be something. It's hourglass, yeah. It's not really hourglass, but almost like hourglass.
I see. But I would defend the authors of the fungi study and say that despite it is not embryogenesis, they could show or they could find a really clear hourglass pattern that is statistically significant. So in that sense, yes, in fungi it has been developed despite fungi don't undergo embryogenesis.
Right. Now, two notes. First, that's a wonderful quotation from Karl Franz Russian textbook.
Uh-huh.
School is Russian. Exactly that
Really?
phrase. Yes.
Nice.
Because it's really very telling story.
Mm-hmm.
And well, in Russia there was a guy called по-моему, Алексей Николаевич Селевцев, Nikolai Alexeyevich Селевцев, who was sort of developed extend the idea of art hacking, of this, I mean, that sort of Ivo Divo of 1930s. And his point, he made several interesting points that you find all the evolution goes due to changes of ontogenesis. And he loved, you know, all embryologists. They loved the words with the Greek roots. You know, that was hourglasses, something like this. I mean, I don't remember all that. But the idea is that there is some core stuff which appeared, which occurred earlier, and then there are modifications just in case, sort of a local adaptations. So what I'm going to is that you have sort of, if you look at evolution, how the embryogenesis occurred. It occurred in the middle stage. And then it was modified in the end, of course, it's clear, but it was also modified in the beginning. I'm speaking about animals. I don't understand really what the story about the genesis of the plant. But the idea is that, well, I already told the idea. So, what about that? What do you think about this explanation? Can it be checked? Or it's just sort of a natural philosophy in style of Carl Baer?
No, I think it could be checked. So first of all, I should say the second work I don't even know. So I would be really, happy if I could get a translation of that work, if it exists in English or in German or in some language that I would be capable of understanding. And well, in terms of checking, I would say let's first look at, at this work again, and then it's only a question of getting expression data, maybe from different developmental stages in different species from different parts of the phylogenetic tree to test it.
But also, the morphology is very telling. For example, you start with animals, it's very clear.
Right.
So the early stages are very different. I mean,
right.
And the final stage, but the middle organogenesis is the same.
Yes.
Yeah, that's what.
So on the morphologic level, I should say, this is something where we could perhaps not help much because we are not experts in this. So what we can only contribute is try to supply insight from the molecular level.
Yeah. And it's very difficult to quantify.
That's right.
Morphology.
That's right.
And
but I'm happy about this great proposal, so thank you very much.
Anyway, more questions? Yes.
Oh, about the Drosophila 24 hours hourglass. It's like it is transition from the egg to larva. And what about the transition from the larva to the fly? It's also a hourglass pattern.
I'm not sure. I think people haven't studied it. So we only took the so to speak, the original hourglass data and took the very same data or the very same developmental stages studied there, and only looked for this expression data. This is not where we did the experiments, right? We don't have a Drosophila lab. This is just the expression data that's publicly available, and you just compute in addition to this tie value, which was computed in the original Nature publication. We only compute now this TDI value or this TDI profile for different stages for exactly the same expression data and exactly the same genes, right? So good question, but we don't know. Maybe by now new data exists that covers other stages of development, and then we should look at this. So I can tell you that we also spent a lot of time making the algorithms available. So there's now an R package that we can provide. It's called MyTie, and it's publicly, we make it publicly available so every one of you can use it. It should be very intuitive. We tested it with several dozen biologists from our area who don't know anything about Ivo Divo or these things, and they could use it. They helped us make it usable, right? Initially they couldn't, they told us where they got stuck, so we improved these things. So if you like to perform these studies on the computational side, it should now be very simple. You should get the expression data you like, and then you can actually go.
Thank you.
Okay, no more questions? Thank you very much, Ivo.
Thank you very much. It's my pleasure. Well, thank you very much. Thank you very much. You're such a great team. And, well, keep up the good spirit, keep up the good work. Have a great evening, and see you next time.
I will give to Victoria the name.
Okay, that's nice. Yeah. I'm happy that you liked it. The talk. Right. Yeah. Oh, that's okay. Yeah, if you like to use some of it, feel free. Hey, it's so nice to see you again. I mean, I promised to you to write you, but of course I didn't. On the other hand, well, but I was, you know, that's another thing I was that it really makes sense. But in fact, I mean, I would strongly like to invite you. There are many people of us, you know, in our university.
Не знаю, у меня уже ушло на Нобелевский премию. Да. Тогда давайте